in the digital age that we are moving forward to, we cannot send our DNA sample to one lab using one instrument, send another sample to do RNA analysis to another lab, the protein to different lab. We need to actually have a platform that can monitor all of these analytes on the same platform, because this will enable us to tap into biology and look at these markers at the same time, which then enable us to really understand biology much better. And also, because our platform does not require optics, does not require labeling, you can, with the same platform, by changing the biology that you anchor to the transistors, you can detect different things, different analytes. You can detect biomarkers that are associated with cancers, like free circulating DNA. You can monitor exosomes, which are these extracellular vesicles that are carrying a lot of information that are used for cancer research and other pathologies. You can look at immune response. You can look at circulating cells, and you can look at volatile organic chemistry and a lot of different analytes within the same platform. And this is really the future. And the beauty of biology is everything in our body is charged. Our proteins are charged molecule. Our DNA is a charged molecule. RNAs are charged. So as this component, anchor and bind to our graphene transistors, we can monitor them in real time. And this is basically the core of how the technology works. We actually anchor different type of biology to our graphene sensors, and we look at the interaction of this biology with our analyte of interest that is found in our blood, in our serum, in any biological sample. And as this binding happens between the anchored biology to our surface and the actual analyte that we're looking for, we can see the signal live with our sensor, because these sensors are extremely fast in monitoring and seeing this electrical signal.
You guys, over the past 8 years, have accumulated this body of knowledge through all the trials and testing that you've done to build that library, because as far as I know, that's not something that you can go and reference. It's not something that you can infer. It's only something that comes from actually testing each of these different biological signals over many, many, many reputations to refine what that signal looks like. Am I missing something? There's no shortcut to that, is there?
Absolutely not. I'll give you an example. For example, with our CRISPR chip, we have tests of 500 transistors to actually compare the result, analyze, understand, and develop a software that can actually detect that. And and our knowledge, as we expanded our partnership with different academic and industry partner and brought people from different disciplines together, was what really enabled us to get the ones that we already feel comfortable with working. And we're really hoping that if there are other markets, for example, with volatile organic chemistry, with our collaboration with GATE, an expert in transmembrane proteins, which can detect the VOCs, we can understand how does the biology is impacted by electronic, and all of those things. It's really important to understand and develop those assays. So it takes a lot of effort and a lot of people from different disciplines to get to a point where we are today in terms of those set of analytes that we can detect. Yes.